All right, so in the lab, we are indeed interested in multi-drug resistance and vaccine escape, especially to explain emergence and evolution of bacterial strains. I will, as Guido kind of guessed, talk about Corynebacteria and Bordetella, and will not talk indeed about Klebsiella, although that's also an interest in the lab, especially in the field of genomic taxonomy developments. So this slide is kind of simple, but just to recap that when we want to characterize the emergence of bacterial pathogens, obviously genome sequencing is now the great way to go because it's all-in-one technology, as we say. So we can not only recapitulate the genotypic background of the strains, but also have access to their what we call accessory genome in bacteria, which are containing resistance genes or virulence genes, including antigens that may correspond to antigens in the vaccines. And so with this approach, we can answer many questions about bacterial evolution and emergence. And today I will focus on international dissemination, as this is kind of, as I understood, a main topic of this colloquium, but also on vaccine escape, because that's an important theme in the group, and also we will see how we can apply and learn from genomic sequencing in the case of outbreak investigations. So that's kind of an outline of my presentation. I'm not completely sure I will be able to talk about all of these topics because I tend to be a bit slower than expected. But there are 4 points. So the first one will be how and why do these bacterial pathogens emerge despite vaccination? Do they escape vaccine-induced immunity in the human population? And how do we measure spatial spread at local to global scales? I will present that and illustrate that on the example of Bordetella pertussis. And then perhaps if we have time, we can discuss current challenges about bacterial genomics and vaccine-preventable diseases. So diphtheria used to be a very important disease historically. Of course, it was the first killer of young children until the turn of the 20th century. So the classical form is a respiratory disease with the typical pseudomembrane that you can see in the back of the throat there. There are other clinical signs like the bull's neck, for example, with laryngeal involvement. And there is a less classically described clinical form, which is the cutaneous form, which I will come back to later. And these 2 primary forms can kind of have complications if the toxin is present. So the diphtheria toxin was the first bacterial toxin discovered, in fact, by Emil von Behring, and I will show that later. And when this toxin is introduced into the bacteria by the prophage, which integrates into the chromosome of the bacteria, then the strain becomes toxigenic. And this can lead to complications during infection, in particular heart complications or renal complications and paralysis. So indeed, diphtheria was a kind of pioneer bacteria in microbiology. Discovery of Corynebacterium diphtheriae, the main causative agent, was actually a German feat with Klebs and Loeffler. I believe Loeffler was a colleague of Robert Koch. And the diphtheria toxin obviously was kind of discovered by Yersin and Roux, at least this is the claim from Institut Pasteur point of view. But on the other hand, a very important discovery was made by Emil von Behring and Kitasato, who were both actually colleagues and mentored by Robert Koch himself, right? You probably know that more than me. And so also one of the first clinical trials ever was made on diphtheria, and also vaccines based on anatoxins, which means the diphtheria toxin, which was detoxified were actually invented using diphtheria as a model, and very quickly after on tetanus. Prophages as vectors of virulence factors, etc., etc. So there is a kind of famous, glorious history on diphtheria in terms of bacteriology research. Now, diphtheria has almost disappeared as a disease, at least in Western countries or Northern countries. You see the red and blue curves rising, which are the vaccine coverage estimates by WHO, and the reported countries' vaccine coverages, but also in blue bars, you see the incidence of diphtheria over the years from 1990 to more recently, 2013. And obviously, that's a nice correlation, and there is a lot of evidence that there is some causal effect behind this correlation. So one of these is that, unfortunately, in many countries, coverage is suboptimal. And so you see the darker red the countries, the lower the vaccination rate. And in squares, you can see the recent outbreaks of diphtheria. For example, in Yemen, in 2017, there was an outbreak, and you can see that the vaccine coverage is suboptimal. We estimate that when the vaccine coverage is below 80%, there is a strong risk of outbreaks. So more recently, there was, and this is actually just ongoing, in Nigeria, Guinea, and other West African countries, an outbreak, a very large outbreak with more than 30,000 people. And in Europe, we had an event. I'm not sure we should call it an outbreak, but we can call it an event among migrant people with nearly 400 cases. In this case, cutaneous diphtheria, and I will come back to that. So the first example I would like to highlight is the diphtheria outbreak in Yemen, because we actually were involved in the reaction to this event by being contacted by the Public Health Institute of Sana'a in Yemen. And so this outbreak was declared in 2017. WHO made a number of statements in the years after, and you can see the numbers, and you can see the epidemic curve here over a few years. And this was one of the largest outbreaks since perhaps the largest outbreak since the big outbreak of diphtheria in the mid-90s in the ex-USSR. So one obvious explanation for this rise in cases was the low vaccine coverage, as we already hinted. Indeed, we collected data from different governorates and showed that based on this data, they were probably much lower than the 80% kind of threshold considered as protective at the population level. And another evidence that vaccines work was that WHO and other public health agencies organized a catch-up vaccination campaign in Yemen. And you can see here on the two periods of the outbreak on the left and on the right, and the red curve corresponds to the proportion of cases in young children below 5, which were the main target of this vaccination catch-up. And you can see that between the first and the second period after the vaccination catch-up, the proportion of these children being affected was much lower. And this is also possibly explained by this vaccination approach. So what can genomics bring actually to this outbreak? At this step, there were almost never a genomic analysis of diphtheria outbreak. And so we were interested to get more knowledge about what was going on from a microbiological point of view. So when we were contacted by the public health lab of Yemen, we answered that we could collaborate and would be very happy to help them. So they managed, and it was not very easy in the context of the civil war, to send us 100 stabs of diphtheria cultures, which were shipped to Institut Pasteur. And from these, we could recover 42 which were still alive in pure culture. And we characterized them by a number of approaches, including genomic sequencing. So here you see a phylogenetic tree. And thank you, Richard, for introducing what our phylogen is. So it's obviously much more reduced in numbers than what we have just seen for SARS-CoV-2 and other viruses. This is probably the other end of the spectrum in terms of volume of sequencing that was performed ever. Anyway, so the first surprise was that you can see diversity within an outbreak. So this was not a monoclonal outbreak, which is typically seen in foodborne pathogens, for example, or nosocomial infections. And this tells us a number of things that I will come back to. The other surprise was that there was a lot of heterogeneity also in the phenotypes of the isolates. You can see all these dots and black squares on the right-hand side of the phylogeny. So the dots are genes that we detected in the genomes, and the squares are phenotypes in terms of antimicrobial resistance. And basically, there was a lot of heterogeneity, for example, for trimethoprim, for ciprofloxacin, for beta-lactam resistance. And also toxin was not always present. There was one lineage that was tox negative, in fact, which is interesting. And also many different siderophores and other valence factors were either present or absent in these strains. And so this tells us that when you have an outbreak of diphtheria, you should probably be aware of this heterogeneity in terms of clinical care and perhaps also public health measures for transmission control. Another teaching of this outbreak was, and here we used similar methods that were just shown on SARS-CoV-2. So using the number of mutations from the tip of the branches, so that's the strains, to the root of the tree, and taking into account the date of sampling, we can infer the mutation rate of the pathogen, which is indicated here on the top left. And based on that, we can also estimate the age of the common ancestors of these different nodes in the tree. And to our surprise, the common ancestor of this sublineage is just one of 6 main lineage that I just showed previously. So for this sublineage, the common ancestor was a bit, maybe 2 or 3 years before actually the recognition of the outbreak, meaning that probably there was already circulation of diphtheria, of Corynebacterium diphtheriae, in Yemen before the outbreak was really recognized. And that's also an interesting learning from this outbreak. So this slide summarizes kind of a number of things that I've said already. Multiple chains of transmission, a silent diversity reservoir must persist, probably, despite vaccination. Otherwise, we would not see all these different genotypic variants popping up when vaccination is dropping. Clearly, there is a need to enhance clinical diagnostics and surveillance to detect outbreaks earlier. And we were very limited in our understanding of the source of these strains because no genomic sequence was available from neighboring countries. And so a more systematic application of genomic sequencing would allow the geographical dynamics of diphtheria to be better defined. Now, a second example of diphtheria outbreak investigation is this European event. So in 2022, we witnessed nearly 400 tox-positive cases in Europe. These were in different countries that you can see on the map and on the slide. Mostly young males, many, probably 2/3, either from Afghanistan or from Syria. No transmission to EU residents. And in almost all cases, it was cutaneous forms. And the cutaneous forms of diphtheria are less known than the classical respiratory form, although this is a very common type of infection in tropical areas. And it probably plays an important role as a reservoir of diversity of diphtheria, even in vaccinated populations. So in our countries, in France, for example, these infections are associated with a very precarious lifestyle. Most of the cases are homeless people, for example. Now, what about genomics of this outbreak? So we looked at the genome sequences of 366 isolates. You see the tree here. So this tree is now circular. So the root is kind of in the middle somewhere here. No, I should show on the, yeah, here for those that are online. And so basically what we see is also a diversity of lineages. You see here, for example, 3 or 4 lineages, but then there are 1, 2, and 3 larger lineages that were sampled very often. And these lineages were sampled in all the countries that were involved. You can see here how the different European countries are intermingled in this tree. And so that's one first conclusion, is that a bit like in Yemen, we observe quite a diversity. It's not a monoclonal outbreak. The other observation, similar to the previous one, was that the strains differed in their complement of resistance genes. In particular, we observed resistance to erythromycin, so macrolide resistance, which unfortunately macrolides are the preferred treatment in many cases to avoid beta-lactam allergy. And so this is concerning, obviously, and that's something that has not been frequently observed. So we witness the emergence of erythromycin resistance in this outbreak. Yeah. All right. So if we look a bit closer at the diversity within these different main genotypes, here, for example, ST384, we see a diversity of strains from the different countries, but not much diversity. So remember, keep in mind, these are bacterial genomes with nearly 2 million base pairs that are sequenced. And we see only the number of mutations that you can observe on the branches here. So I believe this is 5 or 3 mutations, or even 2 perhaps. Yes, 2 mutations, so very limited genetic diversity within the main sublineages that were observed, meaning that this is clearly a common ancestor, very recent, probably 1 or 2 years, no more. And we have estimated that in some cases. Interestingly, this ST was also connected to the Yemen outbreak, but much more distantly, perhaps a few dozens SNPs or mutations, meaning that there is no direct connection between Yemen and the European outbreak, but still it's interesting to see that one of these genotypes that we saw in Europe was also previously observed in Yemen. So clearly what the genetic similarity between these isolates tell us is that there was transmission during this event, and we don't know if this was during the routes of migration or after migration within the centers where the migrants people sometimes gather. So that's unknown due to the difficulty in epidemiological investigations of these populations, but there are a few detailed studies from Switzerland and Germany on this outbreak at the local level. So my conclusions from this second part are that tox-positive diphtheria are diverse within outbreaks, and this diversity should be taken into account also for antibiotic treatment in particular. Genomics suggests transmission along migration routes. Macrolide and beta-lactam resistance are emerging. I didn't show beta-lactam resistance, but there were indeed a few strains with beta-lactam resistance genes. Yes, and obviously these migrants population, so we didn't have the precise evidence for that, but most of them were interrogated, and in the cases where it was the information was available, the patients had no vaccination history. So, I mean, they were not vaccinated in most of the cases, although this is really not a very strong information certainty. All right, and so this is something that I think we will also discuss tomorrow during the general discussion, that there is clearly, I mean, this outbreak illustrates how the lack of prevention and care in these vulnerable populations also creates threats, first of all, for them, but also perhaps for the hosting populations and the clinical care population, etc. Oops, something's happening. Yes, I have videos of whooping cough. So I'm turning now to whooping cough. So it's kind of a hard transition. It's a kind of third story of my 4 main points. So whooping cough is also called pertussis. The agent is Bordetella pertussis, and there is a minor agent called Bordetella parapertussis, but we are not going to talk about it today. Respiratory droplet transmission, paroxysmal cough, these are all classically known. And an estimation from WHO a few years ago was 24 million cases annually in children less than 5 years and 160,000 deaths. It's a human-only pathogen like diphtheria that I didn't mention. Okay, so one concern with pertussis, and a bit like we saw with RSV and other pathogens, pertussis was disturbed. The natural epidemiology was disturbed during SARS CoV-2 due to control measures. But on the other hand, there was, and so currently we are really witnessing a resurgence of pertussis since a few months in many European countries, and not only in Europe. There was anyway a trend for resurgence that is observed in many northern countries since perhaps 20 years. You can see all these curves here. Typically, the curves go from 1980 to 2012. So it's 30 years, and you can see that the trend is growing in many countries, including in the CDC, in the USA, data from CDC. And here you have a small indication of Tdap. So that's the introduction of the acellular pertussis vaccine. And many persons believe that this played a role in the resurgence of this disease. So what are the possible explanations? The change of vaccine is one. Also, there are suspicions that in some cases better diagnostics, like the introduction of qPCR, has also contributed to this apparent increase. But also, and that's something we are more interested in as microbiologists, partial strain vaccine escape might play a role, and I will detail that in this presentation. So the vaccine shift. So since the 50s, there was a very good vaccine, which was called a whole cell vaccine against pertussis. It was good in terms of efficacy. But it was difficult to reproduce from batch to batch. There were many variations, even in a single country. Not every country used the same strain. There were also issues sometimes with batches in production. But also, these vaccines were at some point suspected to be too reactogenic. And there was a kind of reaction against these vaccines being used in young children. And so, around 95, but depending on the different countries, it's a bit variable. Acellular vaccines were introduced, meaning based on a number of antigens or toxins of Bordetella pertussis that were purified and combined in a single vaccine. These antigens are the pertussis toxin, FHA, so filamentous hemagglutinin, and 2 fimbrias called FIM2 and FIM3, and one other surface protein called protactin, or PRN. And among all these antigens, so all are believed to be virulence factors, but only protactin is really attached to the surface, to the cell wall. The others are either protruding fimbriae or FHA, or toxins that are released in the extracellular medium. And so this is important because antibodies against all these different proteins might be able to block the effect of these virulence factors, but in terms of survival of the bacteria, probably protactin only has an effect because it will induce opsonophagocytosis, perhaps. And so that's something interesting to keep in mind. Now, what happened with this protactin antigen in Bordetella populations? So this is a study we performed recently, and there was prior evidence from different countries that protactin deficient Bordetella pertussis strains are emerging. This is the case in the US and Australia, for example, since the mid-2000s. And in Europe, if you consider the date of introduction of Pertactin, of acellular vaccines, and the proportion of Bordetella strains that don't express Pertactin, you see there is an interesting correlation. And so this is strong evidence that Pertactin is selected against in populations where acellular vaccines, which contain Pertactin, are used. And another evidence is phylogenetic evidence that this is data from France, but there are many other evidence now. Pertactin has been kind of disrupted in Bordetella pertussis by many different genetic events independently in the phylogeny, as you can see here in red. And this is also strong evidence of parallelism in the evolution of Bordetella, highlighting a very strong fitness advantage of being Pertactin deficient. So that's probably a very, very strong case. So clearly there is evidence that being Pertactin positive as a Bordetella is not a very good idea if the human population is vaccinated with acellular vaccine containing Pertactin. So together with my colleagues Henrik Sallier and Noémie Lefranc, we were interested to define quantitatively kind of the fitness of the different strains and the different genotypes of Bordetella depending on the vaccine, the type of vaccine used in the different countries. So here you have to explain a bit this tree. The root is here in the center. You have a genotype called PTXP1. So this is a promoter sequence of the pertussis toxin. There is a variant called PTXP1 and another one called PTXP3 due to one SNP in this promoter region. So this defines two different genotypes. And PTXP1, as you can see, is the ancestor. And PTXP3 appeared kind of in the 60s, according to this timed phylogeny. Now, we have a second important landmark mutation called FIMS3-2, which appeared actually here. So within the PTXP3 genotype, you have a subdivision of two other genotypes. One is FIMS3-1, that's the allele that is ancestral. It was also already there. And FIMS3-2 is a non-synonymous mutation that appeared in this Fimbriae tree at this point in time, so probably in the 90s or early 20s. And on top of these two landmark mutations, we have Pertactin. This is the external circle. And when Pertactin is mutated or disrupted, it's highlighted in black, I believe. And so you see we have six different possible combinations of genotypes. And we were interested in measuring the fitness, depending on whole cell vaccine or acellular vaccine use in the countries, of these six different genotypes. So for that, we used a model that Henrik Sallier developed together with our PhD student, Noémie Lefranc. And we took into account, so this is a logistic model that I'm kind of unable to explain to you. But we tried, questions should be addressed to Henrik and Noémie. But we tried to fit the change in frequencies of these different genotypes according to the genomic sequences, so the genotypes, but also according to the introduction of the two different vaccines in the populations. In blue, you see the era where for each country, the whole cell vaccine was used, and then the acellular vaccine in orange. So using all this data, Henrik and Noémie were able to measure the fitness of all these different combinations of genotypes. So here you see PTXP1, PTXP3, and here PTXP3 FIM2. So these are the three main subdivisions of the phylogeny. And for each of them, you have either Pertactin positive or Pertactin negative isolates. And for each of these, you have the whole cell vaccine era and the acellular vaccine era. And here you see that there are many sharp differences in fitness between the different eras. For example, for the pertactin positives, here you see a drop, here you see a drop, here you see a drop. So in the acellular vaccine era, clearly being pertactin positive is a disadvantage, whatever the genotype. But if you are PRN negative, there is also a disadvantage here, but here a strong advantage in this genotypic background. And here we summarize on the right the different relative fitness of being pertactin positive or negative according to the vaccine era. And you see here a big jump in fitness for pertactin negatives in the FIM3-1 background. So that's quite impressive, and we have very big fitness differences between these different genotypes, and especially if pertactin is present or absent. And as you can see, the effect is different according to the vaccine era. Okay, so I guess I have shown you that the population evolution of Bordetella pertussis occurred by allelic drift. So I've not talked much about the evolution of the pertussis toxin, but there is also some variation in this antigen, etc. And that we have clear signatures of vaccine-driven evolution for pertactin. This is really a kind of textbook model for this kind of themes. Now, the questions that arise from these observations are, first, what will be the impact on vaccine effectiveness of losing pertactin? This is something that's of high concern for vaccine manufacturers, because pertactin was really clearly probably the most reactogenic antigen in the most antigenic antigen in these vaccines. Also, there are interesting questions about the severity of disease by strains that don't express pertactin, because this is a known virulence factor of Bordetella pertussis. And all these data have clear relevance for future vaccine design, and especially the perspective of defining vaccines against colonization rather than disease, as are currently designed these vaccines. Now, I have an entire section on the spatial spread of Bordetella, but I was kind of asked to speak during 35 minutes, was it? So we are done almost. So you know what, I will skip this entirely so that we don't have frustrations. But basically, we quantified the speed at which the populations of Bordetella pertussis equalize over time across different spatial scales, from regions within France to different countries in Europe or different states in the US, or between the USA and Europe. And just to give you a sense of this, in 5 years, you have kind of the same populations in the different countries of Europe. In other terms, if you take two strains that diverged during 5 years, they have a common ancestors five years ago, you cannot predict if they come from the same country or from different countries. Interesting. So we can discuss that more, because I'm afraid this was a bit too ambitious. Okay, so vaccine preventable pathogen genomics. So some conclusions, perhaps, to wrap up. So the genomics helps understanding emergence and outbreaks and transmission routes, as I've shown for Corynebacterium diphtheriae. It helps defining strain heterogeneity, which is very important to take into account in terms of clinical response or public health response. There are high-risk lineages, and their surveillance should be enhanced. This is something I didn't really highlight during this presentation. And also, this genomic approach obviously raises important knowledge or enhanced knowledge on the population evolution and vaccine strategy challenges. There are many challenges like data sharing, and we have seen great examples in the previous talk of data sharing platforms. And to many extents, the challenges are the same for bacterial genomics. And for diphtheria particularly, but also for pertussis, capacity building and really enhancing the microbiology capacity, but also the genomic capacity at the global level is very important, because it's lagging behind the capacity we have for many other pathogens, including bacterial pathogens. This is an example of the map. If you look at the map, the number of sequences from Corynebacterium diphtheria that are currently available in public databases. And you see, for example, Africa is almost completely gray, meaning there are no sequences. So we cannot understand many aspects of international spread if we don't have any background data from these countries. And this was my slide on Klebsiella, just to please Guido, our chairman. Just to mention that in the case of Klebsiella, which is in terms of bacterial genomics much more advanced than Bordetella and diphtheria, we have now created a unique genomic platform at the global level where we concentrate all the strains, all the genomes sequences from this pathogen. And we have developed harmonized analytical tools that are well adopted by the community, and then reporting forms, etc. So little by little, the community converges on ways to share data, but also analyze them in a common way. And this is a very powerful approach. And this is led by Kat Holt, but also with the help of David Ananson with Pathogen Watch at Oxford. Just allow me to thank a few of the colleagues who contributed to what I have presented you. So first of all, the colleagues from Yemen who worked in very harsh conditions, especially Abdullah Al-Azhari, who is the head of the bacteriology lab at an NCPHL in Yemen, and our two colleagues, epidemiologists and microbiologists. For diphtheria, I should also thank a number of colleagues, in particular for the European study, colleagues from ECDC, Andreas Hofer and Daniel Palm, who Andreas Hofer has really been the leading force behind the study that I presented you, and also Adrian Egli and Elena Seth-Smith in Switzerland. And yeah. And for pertussis, just to mention the important or even critical role of Henrik Sallier in all the modeling studies that I've presented you for pertussis genomics. And this is my last slide highlighting what Guido just presented you. If you are frustrated not to have heard about Klebsiella, you can come to Paris Institut Pasteur in November, and we'll be happy to host you. There are some seats still available. But be careful, it's filling in very quickly. Thank you very much for your attention.
Thank you very much, Sylvain Brice. It's good to know since I have not registered myself. So, okay. So, are there any questions? There's one over there.
Thank you very much for your presentation. I'm Lena Schneider from RKI. And I think it's really interesting to see also the wrap-up of the 2022 outbreak that we observed in Germany and in Europe. Mm-hmm. And of course, here in Germany, it's actually not over. So we also have quite a lot of cases still in 2023. But as you presented on the Yemen outbreak and on this heterogeneity of strains, I wondered about your assessment of the relevance of non-tox diphtheria, because it's not notifiable in Germany. I think it's not notifiable in most countries. So surveillance is really, I mean, we actually don't know that much because we don't have that many isolates. And I would really be interested in your perspective also on if we do assume it's maybe a problem, what do we do then? What's the recommendation if vaccination is not an option?
Yeah, thank you very much. So I think we have to be a bit careful about the presence of tox-negative isolates in Yemen, in the Yemen outbreak. First of all, it could be a kind of co-infection where only one of the two strains was picked for microbiology studies, but we cannot exclude that there was a tox-positive strain in the clinical sample, which was not grown. in France, we have also described a number of tox-negative infections with pseudomembranes in the throat. So it's not completely clear if the toxin is really needed. I mean, to trigger the pseudomembrane symptom. But I think tox-negative strains induce much less virulent infections. This is very clear, and we have never seen severe infections except the invasive ones with tox-negatives. So I don't think we have to update the guidelines based on the presence of, it's a kind of anecdotal presence, I would say, of tox-negatives during diphtheria outbreaks.
Okay. There's a question over there.
Hi, I'm Teresa Tomaszewska from Robert Koch Institute, and I wanted to ask regarding the pertussis part that you showed. There was something interesting I observed on the map that you showed regarding cellular and acellular vaccines. There are, for example, in Europe, there was one country, Poland, which was the only one using cellular vaccines, surrounded by all other countries using acellular one. And on this kind of border countries where or regions where there is a difference between cellular and acellular vaccine, what impact does it have? What recommendations is it based on? And is it known in case it turns out that the acellular vaccine becomes less efficient? Can we simply come back to the cellular vaccine, or does the protracting resistance or lack of protracting influence also the efficacy of the cellular vaccines?
Okay, so there are many questions. Yes.
Sorry.
I guess the last one is easier to answer. So in countries where the whole cell vaccine is used, the fact, I mean, being pertactin negative doesn't bring a very strong fitness advantage. So I don't think we should worry too much about the circulation of these strains in these countries where the whole cell vaccine is used. So Poland is an interesting country indeed, because it's surrounded by acellular vaccinating countries and still uses whole cell vaccines. We don't have that many genomes from Poland, and they were not included in our common study. But I think I remember a number of publications describing pertactin negative isolates also in Poland, which means that probably these were kind of selected by acellular vaccinated people infections, but then transferred, I mean, transmitted to people in Poland simply, even without the selective pressure itself in Poland, which tells us that there is interconnection between the vaccine strategies of different countries and the countries themselves. Yeah.
Thanks a lot.
Question over there.
Hi, just a quick question about, sorry, about, you said, you mentioned like the importance of surveillance and genomic monitoring of bacteria in general, about these pathogens. I was wondering, now that Paris is hosting the Olympics in the coming months, whether you have any measures in place to monitor the situation before, during, and after.
Well, yeah, so I'm probably not the right person to ask this question, right? But I know there are, obviously, Public Health France and other actors at the Public Health Ministry are busy with it, preparing to the Olympics in their way. And yes, like in many other previous Olympics or big gatherings, there have been examples of outbreaks during the Olympics. So in France, we have different public health systems than in Germany, for example. So Public Health France mandates different laboratories across France to do the microbiology surveillance. Like in Pasteur, for example, we have 14 different reference centers, including the 2 that are in my lab. But and so there are discussions between this network of laboratories and Public Health France to get ready for to be more reactive during the Paris Olympics. But I don't have a lot of specific information on that. One particular concern for us is really whooping cough, because it's really coming back now very, very strongly. I think Taylor Swift herself was incriminated because during her concerts, there are apparently a lot of SARS-CoV-2 circulation, but also probably whooping cough. And so, yeah, it could also happen during the Olympics that whooping cough could even affect competitors. So that might be a big issue.
I may add a personal question myself. So you shortly talked about antimicrobial resistance in diphtheria. So my question would be, how is the situation in relation to other pathogens, where we know that in Arab and in Asian countries, resistance rates are much higher for many pathogens than, for instance, in Central Europe. So how's the situation in diphtheria? And you have not talked about pertussis at all. Is there any resistance issue in pertussis?
Okay. So first for diphtheria, it was known for a long time that there is resistance circulating in Corynebacterium diphtheriae, in particular for tetracycline, trimethoprim and sul. But for the clinically relevant antibiotics like beta-lactams, so far, until a few years ago, there was absolutely no evidence of resistance. For erythromycin, so macrolides, there is this RMX gene that has been described in Indonesia, I believe. But the global map of these different genes is poorly known for the reasons that I've just discussed, that there is no surveillance of diphtheria in many countries. Microbiology is almost not done. We can see it for the African outbreaks that are ongoing. There are currently no genomic sequence in GeneBank from these outbreaks after almost 2 years of outbreak. So it's very unlike SARS-CoV-2, it's incredibly distinct. It's kind of opposite situation. And so what I believe we are seeing at the moment is, because we have got a number of imported cases in France from Africa, from Western Africa last year. And we have described 10 isolates which were multi-drug resistant, which is highly concerning. And it was a kind of surprise, but it looks like in West Africa, the Corynebacterium diphtheria strains are very frequently resistant to beta-lactams, penicillin in particular. And for that reason, now WHO has issued a new guidance document for clinical care of diphtheria in Africa, recommending macrolides as a first line. And knowing that macrolide resistance genes are circulating, this is also a big concern. In other world regions, very difficult to answer. For diphtheria, we have almost no data. And by the way, we have recently worked with the UCAST group to define breakpoints for the agents of diphtheria, so Corynebacterium diphtheria, but also Corynebacterium ulcerans. And this may help to harmonize a bit the way the different publications report resistance in diphtheria. Now for Bordetella, very concerningly, in China, since like 10 years, we have seen a lot of macrolide resistance. So azithromycin is the preferred treatment for pertussis. And there is a very high rate of resistance to macrolides in China, up to 70–80% in some reports. Very concerning. And to our surprise, based on what I showed you and the quick dissemination of strains across the world, we have not seen macrolide-resistant isolates almost in Europe so far. One was described in 2011 in France, just one isolate, which never spread apparently because it was never seen again. But this year, with the big resurgence of Bordetella pertussis, we have already seen 2 macrolide-resistant isolates. This is a kind of scoop. It's not published. Only Santé Publique France knows it, so now you are the second ones to know.
So now we know also.
But this is of concern, of course, and we need to be very careful about the spread of these isolates, because the clinical response in babies is very different with the macrolide-resistant Bordetella pertussis.
Okay.